A blooming flower is an active, mechanical process. It is a dynamic structure opening to expose a system optimized for sexual reproduction. In strict botanical terms, the flower is a modified reproductive shoot. It is the site where the plant transitions from vegetative growth to specialized cellular development. The biological objective is the formation of seeds and fruit. Every part of the flower exists to facilitate the fusion of gametes to achieve this goal. This longitudinal section reveals the macroarchitecture, which is organized into 4 specific whorls or layers. The calyx and corolla are the non-essential or accessory whorls. The green sepals provide structural protection during development, while the petals serve to attract pollinators. Direct reproduction occurs in the essential whorls, the andrecium, or male stamen, and the gynoecium, or female pistil. This is where gamete production takes place. These whorls sit upon a swollen foundation called the thalamus, supported by the pedicel, which acts as the flower's stalk. To understand the production of a seed, we must analyze the pre-fertilization phase, the period where the flower builds parallel factories to manufacture male and female gametes. The androcium is comprised of stamens. Each consists of a slender filament and a terminal anther. A transverse section shows it is typically bilobed and dificus, meaning each lobe contains 2 chambers. This creates a tetrasporangiate structure with 4 circular microsporangia. These are surrounded by a 4-layered wall for protection and nutrient transport. The outermost layers, epidermis, endothecium, and middle layers, protect the internal tissue. They also facilitate dehiscence, the physical rupture releasing pollen. The innermost layer is the tapetum. Its cells have dense cytoplasm and are often multinucleate, providing nutrition for developing microspores. Inside the microsporangium, diploid pollen mother cells undergo meiosis. This process, Microsporogenesis, produces a cluster of 4 haploid cells called a microspore tetrad. As the anther matures and loses moisture, these tetrads separate into individual pollen grains. A mature pollen grain is organized with a specific anatomy for survival. The hard outer layer is the exine, composed of sporopollenin. It's one of the most resistant organic materials known. Protecting the grain from extreme conditions, the exine is absent at the germ pores. Beneath is the thin intine layer. Internally, it contains a large vegetative cell for storage and a smaller generative cell that produces male gametes. In 60% of angiosperms, pollen is shed at this 2-celled stage. In others, the generative cell divides into 2 male gametes before release. While these grains can remain viable for months, they are also frequent triggers for respiratory allergies, such as those caused by Parthenium grass. The male system functions as a high-volume factory, producing pollen grains that utilize multiple protective layers and sporopollenin to survive the transit between flowers. The female reproductive organ is the pistil. It is composed of the receptive stigma, the elongated style, and the basal swollen ovary. Within the ovary, the placenta serves as the attachment point for the ovules. Each ovule is connected by a stalk called the funicle, joining the body of the ovule at the hilum. A typical anatropous ovule is encased in protective integuments, which leave a small opening called the micropyle. Inside is the nucellus, a mass of cells storing food for the developing gametophyte. Megasporogenesis begins when a single megaspore mother cell in the nucellus undergoes meiosis, resulting in 4 haploid megaspores. In most plants, 3 of these degenerate, leaving only 1 functional megaspore. The nucleus of this functional megaspore undergoes 3 rounds of free nuclear mitosis. This produces 8 distinct nuclei floating within the same cell membrane before any cell walls are formed. Once cell walls develop, it organizes into a 7-celled, 8-nucleate embryo sac. At the chalazal end are 3 antipodal cells. The center is occupied by the large central cell, containing 2 polar nuclei. At the micropylar end is the egg apparatus, consisting of 1 egg cell and 2 synergids. The synergids feature a filiform apparatus, specialized structures that guide the pollen tube to the egg. The female system concentrates resources into a single, precisely organized docking tay. Protected deep within the ovule, it awaits the arrival of male gametes. Pollination serves as the bridge between these 2 systems, transporting pollen grains across the physical gap to reach the stigma. Upon landing, a chemical dialogue occurs between the pollen and the pistil. This interaction ensures that only compatible pollen is allowed to germinate. If compatible, the vegetative cell forms a pollen tube that grows down through the tissue of the style toward the ovary. The tube enters the ovule through the micropyle and penetrates a synergid. Here, it releases 2 male gametes into the cytoplasm of the embryo sac. In the first fertilization event, syngamy, 1 male gamete fuses with the egg cell to form a diploid zygote. Simultaneously, the second male gamete fuses with the 2 polar nuclei in the central cell. This triple fusion results in a triploid primary endosperm nucleus. Endosperm development must occur before the embryo develops. This sequence ensures that the zygote has a dedicated nutrient supply to support its growth. The zygote then undergoes embryogeny, passing through globular and heart-shaped stages to become a mature embryo. Following fertilization, the ovules develop into seeds as the protective integuments harden. The ovary itself ripens and expands to become the fruit. While this is the standard cycle, exceptions such as apomixis, seed production without fertilization, and polyembryony, the presence of multiple embryos, demonstrate the flexibility of these reproductive systems. The specific physical organization and nuclear divisions of the flower serve one fleeting purpose: total transformation. This entire system exists to ensure the plant's genetic code survives within the protective casing of a seed.